Welcome to the deep dive. Today we're looking at something, well, potentially game-changing for environmental safety. It's about spotting harmful chemicals in water, but way, way faster than we usually can. We're diving into a technique called dielectrophoretic field-flow-fractionation, bit of a mouthful. Let's call it dFFF. And it uses cultured human cells, HL60 cells, in a clever, contact-free system.
Yeah, and it tackles a real bottleneck. Standard chemical tests, they tell you if a chemical is present, maybe how much, but they don't tell you the biological impact. Is it actually harming living cells?
Right. And the alternative, these in vitro cell tests.
Well, they're often slow. They typically look for gross damage, like the cell membrane breaking down completely. That can take hours, sometimes days.
You could miss the crucial early warning signs.
Exactly. The whole point of dFFF is to catch those very subtle, very early changes in the cells much sooner.
Okay, let's get into the mechanics then. You said contact-free. How does that work? And what's this fractionation thing about?
Contact-free just means we don't need to stain or kill the cells to measure them. They stay live in suspension. The technique itself, field-flow-fractionation, is basically a way to separate particles or cells. In dFFF, it uses tiny electrodes to create specific electrical fields. That's the dielectrophoresis or DEP part.
Okay, electrical fields acting on the cells.
Precisely. And what it's measuring are tiny changes in the cell's outer layer, the plasma membrane, specifically its electrical properties, capacitance and conductivity.
And why focus on those electrical properties? How does measuring the cell's sort of electrical skin tell you more than waiting for it to, die?
Because those properties, capacitance and conductivity, they change very early on when a cell gets stressed. Think of it like detecting a change in heart rhythm before a major cardiac event. It reflects shifts in the cell's metabolism or internal signaling. These changes happen long before the membrane gets physically damaged, which is what the older viability tests wait for.
So the electrical field interacts differently with stressed cells versus healthy ones.
Exactly. The DEP force pushes on the cells differently based on their electrical state. This changes how fast they travel through the system. We measure this change in speed, or elution time, and quantify it using an index called DFS. That's our signal that something's up.
Okay, so it's not just a bit faster. It sounds like it's detecting things on a whole different level. What happened when they tested actual pollutants?
The results were, frankly, pretty dramatic. They tested 8 different toxicants, things like acrylonitrile, endosulfan, paraquat, and for almost all of them, dFFF detected a significant response really quickly. We're talking within the first 150 minutes of exposure.
150 minutes? That's incredibly fast compared to days. But what about sensitivity? Could it detect very low concentrations?
That's where it gets really startling. Compared to the standard cell viability assay, well, for some agents, the difference was huge. For N-nitroso-Nmethylurea, NMU, and also for paraquat dichloride, dFFF was up to 100,000 times more sensitive.
Wait, $105? 5 orders of magnitude.
Yes, 105.55 times, able to detect effects in far, far lower concentrations within that initial 150-minute window.
That's astounding. What about something like endosulfan?
Endosulfan is a mitochondrial poison. And even there, dFFF was slightly more sensitive than a specialized test, looking just at mitochondrial potential. And compared to the general cell viability test, dFFF was still 30 times more effective for endosulfan.
Okay, incredible speed, incredible sensitivity, but no technique is perfect. Where does dFFF maybe fall short?
Good question. There were a couple of areas. First, it wasn't great at picking up direct DNA damage. When they tested something like actinomycin D, which directly messes with DNA, dFFF didn't show much sensitivity compared to, say, the standard comet assay used for DNA damage.
So the link between DNA damage and immediate membrane electrical changes isn't always strong enough for this method?
It seems that way, yes.
Right.
The membrane effects might happen later or be too subtle in those cases.
Okay, what else? You mentioned activating enzymes earlier.
Right, the second limitation. Take carbon tetrachloride. It needs an enzyme, cytochrome P452E1 or CYP2E1, to become toxic inside the cell. The HL60 cells they used, they don't have much, if any, CYP2E1.
So the dFFF didn't detect much toxicity because the cells couldn't actually activate the carbon tet.
Exactly. The raw chemical wasn't doing much harm without that metabolic step.
That sounds like a potential hurdle for broad application, doesn't it? Unless you test every chemical with multiple cell types.
Well, the researchers suggested a pretty neat solution. They proposed co-culturing the HL60 cells with hepatocytes' liver cells.
Ah, because liver cells do have those CYP enzymes.
Precisely. So you'd potentially get the metabolic activation from the hepatocytes and the rapid detection from the HL60 cells using dFFF all in one system.
Clever. And did they show this could work with like real environmental water samples, not just lab chemical?
They did. They took actual canal water, spiked it with endosulfan, and tested it. And the dFFF system maintained its sensitivity. It could still detect the endosulfan effectively, even in that more complex real-world water matrix.
Okay, so pulling this all together, what's the big picture here?
So what we have with dFFF is potentially a very rapid, highly sensitive screening tool. It can analyze, over 100,000 individual cells in just minutes. It really shines at detecting toxic effects that impact the cell membrane or metabolism, often much faster and at lower doses than our current standard methods.
It certainly sounds impressive. So here's something for you, the listener, to consider. If this technology holds up with this kind of speed and sensitivity, how quickly could putting dFFF into routine environmental monitoring change how we detect chemical threats in our water globally? What would that early warning enable?